Okay, so let's continue here. This amino acid, okay, needs to, the ATP is going to bind with the amino acid, okay, amino acid in the presence of amino acid synthetase and magnesium ion. Okay, this enzyme catalyze the binding of the ATP with the amino acid. Okay, so which ultimately forms AAAMP enzyme complex. Okay, so we can say it is an amino acid adenylate complex, amino acid adenylate enzyme complex. Okay, this is what amino acid adenylate enzyme complex. Okay, you please write this term, which is important. Amino acid adenylate enzyme complex which gets developed. So it means that amino acids are get now activated, powered. Okay, now after this what happens, as amino acid now activated, now the second point is that these amino acids, activated amino acids needs to get bind with the tRNA. Okay, because tRNA are actually the adapter molecule which are going to take this amino acids from the cytoplasm into the ribosome. That is why the amino acids are needed to be bind with the tRNA. This is also known as the charging of tRNA. Okay, so as you know, tRNA topic we have already done. If you remember, at the 3' end of the tRNA, where the sequence was CCA or ACC, you can say, where the amino acid actually get binds at the 3' end of the tRNA. Okay, and that is, and one more site is there are 4 arms, if you remember. Okay, and at the DHU arm, the enzyme amino acid synthetase get binds. Okay, there is an anticodon arm. Okay, so at the ACC sequence, okay, where the amino acid actually going to bind. Okay, and this is what is known as amino acid synthetase. Oh, sorry, adapter molecule, the tRNA act as, and in the binding of the amino acid to this ACC sequence. Okay, binding of the amino acid, this substance actually, this is the amino acid adenylate complex. This substance going to bind with the ACC sequence of the tRNA. Here also this enzyme, okay, going to play, or you can say catalyze or trigger the binding of this complex with this ACC, means that of tRNA. Okay, so once this substance get bind with the tRNA with the help of this enzyme, now it is the tRNA is also gets charged. It means the amino acids already get bind with the tRNA. Okay, now this structure, okay, this you can say that amino acid tRNA. So once this substance get binds with it, we can use the term amino acid tRNA. This is the term we can use, amino acid tRNA. Okay. Now this amino acid tRNA, okay, substance, okay, the tRNA actually carrying the entire substance tRNA along with the mRNA going tRNA, the amino acids, is going to enter inside the ribosome through this direction. Okay, this is the entry point. Now you see, first it is the A site. Okay, so this amino acid tRNA, okay, first comes at this site. Okay, I am denoting that complex with the help of this green color. Okay, the tRNA, I am just showing a simple, just to make you understand. The tRNA comes with the amino acid as amino acid at this A site. Okay, it binds here. Now what happens is that from this A site, okay, this combined structure needs to come at the P site. Okay, and you can see here is the codon AUG is here. Okay, once the tRNA come here, let me again draw a basic structure. Here it is the amino acid here. Okay, see, at this side, this is the side anticodon arm, if you remember. Okay, one loop is there also. Okay, here anticodons were there. Okay, so let me just draw it with little more clarity. Just give me a second. Okay, the amino acid, this is the site for anticodon loop. So you see, it is the AUG codon, initiator codon. So here it will be UAC, okay, anticodon, where then it will be binding with base pairing, you can say. Okay, it will be base paired here, codon with the anticodon side of the tRNA. Okay, once it is done, one more thing, remember, at this P side already one enzyme gets bind, is the peptidyl enzyme. Peptidyl enzyme is already there at this side. What its function, that we will discuss. So once tRNA get binds here with this initiator codon here, now this amino acid will be separated, okay, from the tRNA. And remember, in the ribosome already the ribosomal RNA is there. Okay, this ribosomal RNA actually helps in separating this amino acid from the tRNA. Why it needs to be separated? Because tRNA is functioning as a transporter, adapter. It takes the amino acid, it brings the amino acid from outside to into the ribosome, where it needs to unload or separate this amino acid from it. It needs to leave the amino acid in the ribosome, and the tRNA will be leaving the ribosome. Here the function of tRNA is done. So this amino acid needs to be separated out from the tRNA. Okay, will remain there in the ribosome. Okay, and it is no more amino acid here. Now the tRNA is going to leave the ribosome. How? Now once this is unloaded, separated from the tRNA, this amino acid. Okay, and if you remember this, and this is the AUG codon. Okay, which will be the first amino acid that brings the tRNA brings inside. This is the methionine, MET, methionine amino acid. Okay, once it is separated, now this tRNA will be unlocked from here. It will come to the E side, and then from the E side it is going to leave the ribosome. Now the most important point comes here. What happens at the time when this tRNA leave the E side? This is the exit point. From here the tRNA leaves the ribosome. Simultaneously, at the same point of time, the unit of the ribosome, okay, move one codon ahead, one codon forward. Okay, it's a rotation. It's not a simple forward. It's a kind of, you can say, it's a rotation and one codon forward movement. Okay, so and this is the known as a translocation. Okay, and this translocation, that is movement of the ribosome one codon ahead on the mRNA strand. Okay, with the help of the enzyme translocase. Okay, so it moves the one codon ahead. So one codon ahead, once when it moves, what happens? Okay, see, one codon means as it moves forward, what will be the structure look like? I'm just drawing a graph just to make you understand. The P side will be coming at the E side. A will be moved forward. A will come here. Now it will be the P side, it will be the E side. If you look at the second diagram I have drawn here, moving forward, one codon ahead. Now the ribosome is here. Now location also changed along with the ribosome. So A now comes here, P here, now E here. Okay, now again at the A side, what will happen? As ARNA, P or A, whatever it is, it moves one codon ahead. Same story will be now again start. Okay, again another tRNA, the next codon, according to the next codon, whichever it is, the amino acids will be taken by the tRNA inside the ribosome. It will come at the A side first, and A to P, and then P to E, and then again going out. Again translocation, one codon ahead on the mRNA strand. Again the next codon, when it comes, according to that the amino acid will be taken inside. Again this will be continued. Okay, it will be continued, and you can see the amino acids are coming one after another according to the codon. Okay, brought by tRNA, and all the amino acids will be here. Okay, getting stored in the ribosome. And now this peptidyl enzyme which were there, the P side, okay, helps in forming the peptide bond, okay, in between the amino acids. Okay, the bond takes place in between the carboxyl group of the first amino acid and the amino group, okay, the amino group of the next amino acid. The carboxyl group of the first amino acid and the amino group of the next amino acid, in between these two, okay, peptide bond will be developed, okay, with the help of peptidyl enzyme. Okay, and once this chain is forming, you can understand it is a polypeptide chain which is getting developed in the ribosome. Now question comes, till when this process will be continued, and when it is going to stop? Okay, this is the question. Now let's assume that this strand continues. Okay, it's a long strand, it is continuing. Now where this is going to stop? Okay, see, in the genetic code we have studied about the stop codons. There are 3 stop codons. What were those? UAA, UAG and UGA. These are the stop codons which do not code for any amino acids. Okay, so as the ribosome moves on the mRNA strand, any one of the stop codon, when it comes at the A site, okay, any one of the codon when it will come at the A site, or another way we can say, when the A site will be reaching, okay, at any of the stop codon area. Okay, suppose this is a site where UAG is there, for example. Okay, as ribosome is moving on the mRNA strand, okay, when this A will be reaching at this site, okay, at the stop codon site, UAA or UAG or UGA, whatever it is, okay, the process of translation or the process of protein formation will stop. Because why? Because see, as we said, as the A site reaches to the codon, okay, the P site also moves to one codon ahead. According to the codon, the amino acids are brought inside the ribosome. But when the A site reaches at the UAG codon or UGA or UAA, whatever it is, these codons, they do not code for any amino acid. Okay, so it means that no tRNA is going to come with the amino acid inside the ribosome. So the chain will stop. The formation of chain will stop there. Okay, that is why the stoppage of the this process of translation. And the polypeptide chain, okay, what will happen? This is what the protein, the polypeptide chain that is formed is the protein. Okay, it is going to leave the ribosome. Okay, it will be there in the cytoplasm now, the protein structure. Okay, so this is how the protein gets developed. That's the polypeptide which is developed, that is the protein only. Okay, so this is how you can understand. Not much enzyme involved here, very less, 1 or 2 enzymes are there, and the mRNA plays very important role because coded information, the codons are there in the mRNA only. According to the codon, the amino acids are brought inside, okay, by the help of the tRNA, and then it is the polypeptide chain which is forming, and it keeps on going, and when the stop codon comes at the A site, automatically the process stops and the polypeptide chain leaves the ribosome. That is what the protein. Okay, so this is the translation mechanism. As much as I tried my level best to make it, to keep it simple, in a simple language, in a simple process. Okay, so whatever you understood, you can make a gist out of it. Okay, it will help you further to keep in mind to understand things. If any question comes in your mind throughout this process, note down the question, we can discuss in the class also. Okay, thank you so much for watching the video, and wait for my next video. Very soon I am going to upload the next video also. Thank you.